study uncovers first evidence of long-term directionality in origination of human mutation, challenging Neo-Darwinism. This is by University of Haifa, today's article on phys.org. A new study by a team of researchers from Israel and Ghana has brought the first evidence of non-random mutation in human genes, challenging a core assumption at the heart of evolutionary theory by showing a long-term directional mutational response to environmental pressure. Livnat from the University of Haifa showed that the rate of generation of the HBS mutation, which protects against malaria, is higher in people from Africa, where malaria is endemic, than in people from Europe, where it is not. For over a century, the leading theory of evolution has been based on random mutations. The results show that HBS mutation is not generated at random, but instead originates preferentially in the gene and in the population where it is of adaptive significance. This is what Professor Livnat said. And he says, unlike other findings on mutational origination, this mutation-specific response to a specific environmental pressure, like malaria, cannot be explained by traditional theories. He said, we hypothesize that evolution is influenced by two sources of information: external information, that is natural selection, and internal information that is accumulated in the genome through the generations and impacts the origination of mutation, said Livnat. Ever since Darwin, we have known that life arose by evolution, but how exactly does evolution, in all its grandeur, mystery, and complexity, happen? For the past century, scientists have assumed that mutations occurred by accident to the genome and that natural selection or the survival of the fittest favors beneficial accidents. The accumulation of these presumed genetic accidents under natural selection over the millennia leads in turn to adaptations from the hawk's sharp eye to the human cardiovascular system. While widely held in the scientific community, this view has always left open fundamental questions, such as the problem of complexity. Can the sequential accumulation of small random changes, each beneficial on its own, lead within the time span available to the evolution of such astonishingly complex and impressive adaptations as we see around us in nature, such as eyes, brains, or wings, where complementary parts interweave into a complex whole? However, the only alternative to the fundamental level conceived of up until now consists of variants of Lamarckism, the idea that organisms can somehow respond directly to their immediate environments with beneficial genetic change. Since Lamarckism has not worked in general, the notion of random mutation remains the prevailing view. In order to distinguish between the random mutation and natural selection explanation and the possibility that non-random mutation is important, Professor Livnat and his lab manager Dr. Daniel Melamed developed a new method for detecting de novo mutations, that is, mutations that arise out of the blue and offspring without being inherited from their parents. In breaking a new accuracy record, their method allowed something not previously possible: counting of de novo mutations for particular points of interest in the genome. They then applied their method to examine the de novo emergence of the human hemoglobin S HBS mutation, perhaps the most well-known point mutation in biology and evolution. HBS provides protection against malaria for people with one copy, but causes sickle cell anemia in those with two. Malaria itself, a vector-borne blood disease, has arguably been the strongest selection pressure acting on humans in the last 10,000 years, often causing more than 1 million deaths per year in Africa in the recent past. HBS is also used as a central example of random mutation and natural selection and evolution. It has been long assumed that to have arisen accidentally in an individual is in sub-Saharan Africa and then spread inside Africa via natural selection until its malaria-protective benefits were balanced out by its sickle cell anemia costs. By examining the de novo origination of HBS, Livnat was able to disentangle for the first time whether the malaria protective mutation arises randomly and spread in Africa only because of selection pressure, or instead whether it could actually be originating de novo more frequently in sub-Saharan Africans, a group that has been subject to intense malaria selection pressure for many generations. If the mutation is random, then it should be equally likely to emerge in both geographical groups. However, if mutation is non-random, then perhaps it would actually emerge from more frequently in Africans. Therefore, there are at least two possible reasons why such a question has not been asked before, Professor Levant explains. He says, first, it had been assumed that mutation is random. Second, even if one had wanted to ask such a question, it would not have been possible with previous methods. Contrary to widely accepted expectations, the results supported the non-random pattern. The HPS mutation originated de novo, not only much faster than expected from random mutation, but also much faster in the population in sub-Saharan Africans as opposed to Europeans, and in the gene, in the beta globin, as opposed to the controlled delta globin gene, where it is of adaptive significance. These results upend the traditional example of random mutation and natural selection, turning it into an example of non-random yet non-Lamarckian mutation. Mutations defy traditional thinking. The results suggest that complex information that is accumulated in the genome through the generations impacts mutations, and therefore mutation-specific organization or origination rates can respond in the long term to specific environmental pressures, explained Professor Livnat. Previous studies, motivated by Lamarckism, only tested for an immediate mutational response to environmental pressures. Mutations may be generated non-randomly in evolution after all, but not in the way previously conceived. We must study the internal information and how it affects mutation, as it opens the door to evolution being a far bigger process than previously conceived, Livnat concluded. Until now, investigators have been limited by technology to measuring mutation rates as averages across many positions in the genome. Overcoming this barrier, the new method developed by Livnat and Melamed allowed the HPS mutation to be the first to have its mutation-specific origination rate measured, opening up new vistas for studies on mutation origination. These studies have the potential to affect not only our fundamental understanding of evolution, but also understanding of diseases that are caused by mutations, namely genetic disease and cancer. The article was accepted in scientific journal Genome Research, and this is by University of Haifa, unvis.org. So you can see that it has to do with the environment, the pressures of the environment, and how that pressures our genomes to change. Isn't that something? So it's not random mutation. Please leave your comments, and thank you for your support. Finally, support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box.